Hi there. This is another lecture in the Integrated Molecular Evolution series. This part is called Infectious Changes to DNA. I've already talked about mutation. Now I'm going to talk about some of the changes to DNA that are caused by infectious organisms or mobile genetic elements. Most of this can be found in Chapter 13 from the book Integrated Molecular Evolution, the second edition. So here are the things I'll be talking about, including viruses, which of course are infectious transposons, which move around within a genome. Sometimes they can move from one genome to another. Introns, which some of which are mobile still. Plasmids, which of course are mobile. Pili, which accomplish movement of genetic material between one cell and another. So many of these are capable of horizontal gene transfer, that is, moving genes from one cell to another cell. For these gene transfers, they can be either intergenomic or intragenomic. And if you look on the upper right, then you can get gene sequences moved from one cell of species 1 into a different cell of species 1. And this is accomplished usually by pili, plasmids, or viruses, but occasionally by some introns and transposons. Intragenomic is when you have horizontal gene transfer, that is, transfer of the genetic elements from one species into a cell of a different species, and that's done usually by plasmids or viruses. On the lower left, you can also have sequences moving around within a genome, either from one gene into a different location of the same gene, or from one gene into another gene, and this is usually done by plasmids, some introns, viruses, and especially by transposons. So here's a really rudimentary phylogenetic tree just to show some of the relationships, and the bacteria diversified, and one of those groups became the starting point for the archaea. They diverged, and one of those groups became the eukarya, but they fused or had endosymbiotic events with other bacteria to become this diverse group of eukarya. There have been quite a few exchanges of genes, so that there's a lot of overlap among these three groups as far as the genes that they have. This has all been by horizontal gene transfer. If you join the viruses in, then they have a lot of common sequences among all three of those groups as well. The predominant theories about where viruses came from was that they are just reduced versions of pathogenic bacteria and possibly other organisms. One theory is that viruses joined together became the bacteria, but that's a dotted line because that has less support in the literature. When plasmids are joined into this scheme, then they have some characteristics that are similar to bacteria and some that are look more like viruses. In fact, some have characteristics of both plasmids and viruses, and I'll talk about that in a later lecture. The links between transposons and viruses is much more firm because they have some of the same genes and some of the same modes of inserting and extracting themselves or excising from the host chromosomes. Finally, if we add introns into this scheme, then many of them have similarities to transposons and retrotransposons. And so the connection between those is also fairly firm as far as their origin. Just a few more notes. All viruses are obligate parasites, and many transfer their DNA into the host genome. And this includes some viruses that cause cancers, as well as some that cause plant tumors and others. And some members of all of the other groups also transfer DNA into the host genome. So these I'm just classifying as infectious organisms and infectious nucleic acids. Here are some examples of viruses, which are essentially infectious nucleic acids. On the left-hand side are naked viruses, that is, they don't have any membrane surrounding them, just the viral proteins protecting the nucleic acids inside. You have both the icosahedral and icosahedral with a head and a tail, and the tail is mainly for injecting the nucleic acids into the bacterium. Tabemoviruses are plant viruses. They're RNA viruses, single-stranded with a protein coat around. And that protein coat actually protects pretty well because these can survive a burning cigarette or a cigar or any tobacco and still are infectious. And then the archaeal viruses down below are sort of strange looking. And they were just discovered, 15 or 20 years ago for the first time. On the right-hand side are enveloped viruses, that is, they carry along with them part of the host membrane, and then they have viral proteins sticking out of the host membrane. And these include things like herpes simplex, influenza A, paramyxoviruses, and smallpox. The influenza A has 8 RNA chromosomes, so not only one chromosome, but 8 of them, all carrying at least one or more genes on each of the chromosomes. You can see the smallpox virus is fairly complex. It's a DNA virus and has been eradicated from the human population, so we hope that one doesn't come back. Bacterial viruses are either RNA viruses or DNA viruses, and the RNA viruses can be either single-stranded or double-stranded, and also the DNA viruses can be either single-stranded or double-stranded. That is what they carry in their capsids. So they first inject, the RNA viruses first inject their RNA into the cell. They then also carry along with them an RNA-dependent RNA polymerase, RDRP. They make viral proteins from the mRNA. If they're single-stranded plus strands, they need to make a plus strand from the minus strand. If they're double-strand, they just make a double-stranded virus particle. Or nucleic acid, which is then encapsulated into the virus particles that have been formed, and then finally lyse the cell. For DNA viruses, they can use the double-stranded DNA or single-stranded DNA to make an mRNA copy through transcription, double-stranded copies through replication, or if in the double-stranded form, they can become lysogenic. That is, they can integrate the DNA into the host chromosome. This doesn't happen with all viruses, but happens with many of them. They can then transcribe directly from the lysogen, mainly to maintain the lysogenic state. At one point in time, then, if the virus or the bacterium is in some sort of stress mode, they will often pop out of the genome and then start into the lytic phase. And the lytic phase for all the DNA viruses is just to form more virus particles by making viral proteins, replicating their chromosomes, encapsulating those chromosomes, and then lysing the cell. To form a lysogen or to integrate into the chromosome, double-stranded DNA viruses are the easiest to talk about. They will form a hybridization with the host chromosome. Then there's a crossover accomplished by a couple of different proteins, and then breakage and religation of those chromosomes to form a prophage, where the provirus is within the host genome. For RNA viruses, they have to go through an extra step, and that is by using their reverse transcriptase. They can make a double-stranded DNA from that and integrate, although some of the viruses actually integrate as an RNA, and then the host systems will then make a DNA copy of that right in the chromosome and integrate that way. When they excise, sometimes they don't do that perfectly, and they leave a bit of an extra piece of the DNA in the chromosome, and this can sometimes cause mutation of the host genes. Here is a specific example of bacteriophage lambda, which is a double-stranded DNA virus. Here's the genome. It's about 48.5 kilobases in length. Has COS sites on the end, which are cohesive ends. These are self-complementary, so you can circularize the chromosome and they bind, the COS-R and COS-L sites bind together. And when it circularizes, a ligase from the host, from the bacterium, seals the ends, and so it's a circle. On the left side of the linear are all of the genes for making new proteins for the head and tail of the bacteriophage. And on the right is the genes for lysis, but also the regulation and replication regions in the middle, or on the right middle, are primarily controlling regions. And I'll be talking about those in a later lecture. But so you have the circle here, and notice that there's an at site, there's an at lambda site, and there's an at B site, which is on the bacterial chromosome. And these are complementary regions which can bind together. And so there's an attachment there, there's a crossover, and then the lambda phage genome is inserted or ligated into the E. coli chromosome between the gal and the bio genes. Here's a little more detail about that. So on the lambda at the top in red, there's a site that is in the form of POP prime, and then on the bacterial chromosome there's a BOB prime. Those are complementary regions, and then you get a crossover in the middle of that and reformation of the chromosome, inserting the lambda genome into the E. coli genome, creating the lambda prophage, which will then remain there. And every time the bacterial chromosome is replicated, the prophage also is replicated. So it's permanently, or at least permanently for a number of replication cycles, remains in the bacterial chromosome. In the case of viruses for eukaryotes, now with a nucleus, there are 7 different types in the Baltimore classification scheme. Class 1 is for double-stranded DNAs, Class 2 single-stranded DNAs, and then 3, 4, and 5 are double-stranded, plus-strand, single-strand, and minus-strand, single-strand viruses, and then 6 and 7 are a single-stranded RNA virus with reverse transcriptase and a double-stranded DNA with reverse transcriptase. And as you can see on the left hand for the RNA viruses, it's similar to those in the bacterial cells that I just talked about, so that you have a scheme where you need to create a single-stranded RNA, which becomes the mRNA, to translate the proteins, as well as using an RNA-dependent RNA polymerase to make new RNA strands, either the plus strands, the minus strands, or the double-stranded RNAs, which then get encapsulated into the viral capsid and released, usually by budding off within a piece of the host membrane, but some viruses also just bud off and don't take a piece of the membrane with them. In the case of Category 1 and Category 2, they either have double-stranded DNA or single-stranded DNA, and they're similar to those in bacteria, where a single-stranded or DNA or the double-stranded are used to make an mRNA, or they are integrated into the chromosome and become a lysogen. X size at a later point in time. But if they're going to make virus particles, then the mRNA goes into the cytosol to make the proteins, and the virus particles are then assembled around the nucleic acids. And then the virus is released either by budding off of a piece of the host membrane or simply by being released by the membrane. In the case of category 6, which is like HIV, a reverse transcriptase, as well as some other part of a polyprotein is released into the cell. Then the reverse transcriptase makes the DNA copy, finally a double-stranded DNA copy. This can then integrate into the chromosome or immediately start making mRNAs, the single-stranded RNAs, and out in the cytosol make the viral proteins, including the polyprotein that includes the reverse transcriptase. And then these can be released from the host cell. In the case of category 7, they carry double-stranded DNA, but with a reverse transcriptase, and actually make the genomic DNA that winds up in the virus particle by using the reverse transcriptase. So here are some details about HIV, category 6 in the Baltimore scheme. This is HIV. On the outer surface, you have the envelope proteins, glycoprotein 120 and glycoprotein 41, which is a transmembrane protein. And then the red there is a part of the host membrane that it carries along with it. Inside of that is a matrix protein surrounding the capsid protein, and then inside of that are 2 copies of the genome, the RNA, a single-stranded RNA, as well as a polyprotein, which contains, part of it is the protease, and then reverse transcriptase, RNase H, and integrase. So here are diagrams of the genomes of HIV-1 and HIV-2, just 2 variants of the HIV virus. And you can see each one contains 5' and 3' long terminal repeats. Remember those long terminal repeats because they also appear in some retrotransposons, which I'll talk about next. But the genome consists of a GAG gene, which then produces the polyprotein that includes the matrix protein, the capsid protein, the nuclear protein, and one other protein, nuclear capsid protein and one other protein. And then a pol gene that makes a polyprotein, which contains the protease, the reverse transcriptase, and the integrase, which I'd already talked about. And then there's the ENV gene, which encodes for the surface proteins, both the surface protein and the transmembrane protein. And then there are several other genes in there, the VIF, VPR, VPU, etc., and those are responsible for some of the infectious and integrative and excision pathways of the virus. Also notice that the TAT and the REV genes are split, and so you have splicing of those in the RNA to make those particular mRNAs to produce those particular proteins, and those are important in the infectious and integrative parts of the life cycle of this virus. So previously I did mention alternative splicing. So this is sort of like what the TAT and REV genes are doing, but this is more detail into some mRNA genes among different other organisms. So you have a standard splicing mechanism where each exon is spliced together with the next and excluding the introns, but you also have cases where exons are split, they're ignored, you have different promoters that are patched together to create different messages, you have different polyadenylation signals that can be alternatively spliced into or out of the mRNA. There's sometimes intron retention where the intron is actually used as part of the message. and then there are times when parts of the intron are split, and also parts of the exons are spliced in or out. So it sort of is a sort of a crossing point between what is really an exon and what's really an intron, depending on how these things are spliced. As a part of showing you how the reverse transcriptases, as well as some other proteins and genes, cross over into one or another type of genetic mobile element or infectious element, I just wanted to go through a bit of the evolutionary history of some of these reverse transcriptases as well as telomerases. So, as you know, reverse transcriptases are RNA-dependent DNA polymerases, but they're also related to RNA-dependent RNA polymerases. So this is a phylogenetic tree that is rooted in RNA-dependent RNA polymerases, but then it shows all of the DNA-dependent or the RNA-dependent DNA polymerases. And that includes HepaDNA viruses here, retroviruses, chylomyviruses, which are in plants, long terminal repeat retrotransposons, non-LTR retrotransposons, mitochondrial retrotransposons, group 2 introns, as well as telomerases going around the circle there. And this shows the commonality in the proteins of 8 different regions of those proteins. Most contain only 7 of the sections, except for the telomerases, which have an extra section at their amino ends. And so if you look at all of these, you can see that the HIV, which is over on the right, the retroviruses, have a very compact structure as far as their genes and their proteins, and then going all the way around to the LTR retrotransposons, which are found in a lot of arthropod species, including Drosophila, and then the non-LTR retrotransposons, which include the line elements or line-like elements, long interspersed nuclear elements. And those, we have a lot of those in our genome, in human genome. We also have signs, which are another type of, instead of being long, they're short, so they're SINE. And then the mitochondrial retrotransposons, group 2 introns, have these reverse transcriptases, and finally, in your cells, you have telomerases, as do all eukaryotic systems. So the primary reason for showing this is just to show that you have a wide variety of these reverse transcriptases that originated from a single group of reverse transcriptases that came out of the RNA-dependent RNA polymerases. They're in animals, they're in plants, they're in mitochondria, they're in viruses, and so quite a common origin for all of these now slightly different reverse transcriptases. Here is a comparison of each of those sections that came out of a paper a few years ago anyway. And so at the bottom you can see the retroviruses like HIV, and then the next section up you can see chylomyviruses, which are plant viruses, and they only infect plants, so those have been transferred somehow into plants. And then you have the LTR retrotransposons in arthropods, HepaDNA viruses, non-LTR retrotransposons such as in us, group 2 introns, which you find a lot in mitochondria as well. And just above that you see some other mitochondrial elements, including one called a plasmid. So you have plasmids and virus-like elements and introns all having these reverse transcriptases. And then finally the telomerases up above, which are in eukaryotic systems on long linear chromosomes. Here is a phylogram of those relationships, again based on the root of an RNA-dependent RNA polymerase. And so you see the telomerases up there, and they're within the clade that includes the non-LTR retrotransposons, which are within the line elements in our genomes, as well as the group 2 introns and other mitochondrial elements that contain those reverse transcriptases, and then another large group down below that includes HepaDNA viruses and then long terminal repeat retrotransposons that are found in arthropods, and then the cholaemoviruses, which infect plants, and finally the retroviruses such as HIV down at the bottom. So a large group of reverse transcriptases in a very diverse group of organisms and viruses. So this is the end of the first part of this discussion of infectious changes and other changes to DNA. And in the next section, I'll talk about transposons and introns in more detail. Thanks.